I would like to thank USB for organizing the symposium and Craig for the kind invitation to talk about some of the things that I'm currently doing at USDA ARS. As the title indicates, I'll be talking about our efforts in the lab to find procedures, to simplify the procedure to measure trypsin inhibitor activity in soy and soy products. Just like Lisa, I am a plant scientist. I'm not a poultry scientist. So most of my talk will be oriented towards plant side. And as you all know, soybean is one of the oldest cultivator crops. It has been domesticated around 11th century BC. And reference to soybeans have been made in the books written about 4,500 years ago. And the history of soybeans, as far as the United States is concerned, is rather recent. Soybeans were brought into the United States sometimes in the 1800s. And in 1903, soybeans were primarily grown as forage crops. And George Washington Carver at Tuskegee Institute conducted extensive amount of research on soybeans. And his discoveries changed the perception of what we think about soybeans now. With no commercial value at the turn of the century, soybeans is the second largest crop in the United States currently. And soybeans have been extensively used in Asian countries as a common food, whereas its adaptation in North America with regard to the food, human consumption is gradually picking up. If you look at the soybean meal that is produced in the United States, the vast majority, greater than 90% of the soybean meal, basically goes into the animal feed, mainly into poultry and swine industry, and lately its use has been gradually increasing in aquaculture. So soybeans are an excellent source of protein. It contains about 40% protein and about 20% oil. And soybean proteins are relatively cheap, and they have excellent amino acid profile. And subsequently, they are considered as a gold standard when you compare to other protein sources. In spite of the high quality of the protein, soybean cannot be directly utilized in animal feed because they contain several anti-nutritional factors. And I have listed some of the anti-nutritional factors in this particular slide. One is the phytate. As we all know, phytate are the major storage form of phosphorus in seeds and in most of the plants. But these phytates can chelate minerals such as calcium, iron, and zinc, and they will interfere with the absorption of these minerals. And the second is protease inhibitors, and we heard quite a bit of that, and Lisa gave a very good, nice background information on the protease inhibitors in soybean seed, as well as generally in plants. And I'll be focusing my talk on protease inhibitors later on. And the other anti-nutritional factor is the lectins. These are the glycoproteins, which are also called as phytohemagglutinin, and they also are not desirable for the nutrition because they interfere with the absorption. And saponins, again, are considered as an anti-nutritional factors. And in addition to protein, soybean also store about 14% carbohydrates. And among the carbohydrates, you have the oligosaccharides. And among the oligosaccharides, the two of the sugars, namely raffinose and stachyose, these compounds are not metabolized by the digestive system in humans and in monogastric animals, mainly because we lack the enzyme alpha-galactosidase, which is responsible for the breaking down of this particular oligosaccharide. Instead, what happens is these oligosaccharides are fermented and resulting in the release of carbon dioxide as flatulence, and this is responsible for the odor problem as well as a poor growth response. Okay, so what are the most important effects of the protease inhibitors? In the case of soybean protease inhibitors, it has been shown they interfere with the digestion. So they bind with the trypsin, the digestive enzyme trypsin and chymotrypsin, and they make them completely inactive. And as a consequence, the animals are not able to digest the food efficiently and results in poor weight gain. And often it causes pancreatic hypertrophy. And this seems to be very common among poultry, even though there is species variation has been observed, especially in swine and things like that. It's not as prominent as in poultry. So Lisa actually told you about the two most important protease inhibitors in soybean seeds. One is the Kuhne's trypsin inhibitor, and the other is the Bowman-Brick protease inhibitor. The Kuhne's trypsin inhibitor is a 21 kilodalton protein, and it's one of the classical enzymes which has been extensively studied biochemically, and we know quite a bit of this structure of this protein, as well as the catalytic function and the reactive site. Arginine site is responsible for the reactive site, and the other protease inhibitor, namely the Bowman-Brick protease inhibitor. These proteases are named after the people who discovered them, and they have been worked out very, very thoroughly. And the protease inhibitor is a small protein. It's a peptide about 8 kilodalton protein, and there are several isozymes of these proteins. And these two proteins together are mainly responsible for the total protease activity in soybean seeds. So at USDA, ARS, my lab is primarily working on modification of the soybean seed composition. And one of the goals of my research program is to eliminate the anti-nutritional factors from soybean seeds. By anti-nutritional factors, I'm talking about the protease inhibitors, namely the trypsin inhibitor and the chymotrypsin inhibitor. And several years ago, we identified naturally occurring mutants in trypsin inhibitor, Kuhne's trypsin inhibitor. In soybean, there are three types of trypsin inhibitor, KTI, KTI1, KTI2, and KTI3. And we have found mutants of KTI1, KTI3. And KTI3 is the major contributor of trypsin inhibitor activity in soybean seed. So if you inactivate the KTI3, you almost lose 50% of the trypsin inhibition activity. And we were also able to develop lines of soybean lines where we have combined the KTI1 mutant and KTI3 mutant and generated a new line which lacks both KTI1 and 3. So recently, with the funding from USB, by gene editing, we were able to obtain mutants in Bowman-Brick protease inhibitor. So this particular mutant line has significantly, almost 70% decrease in Bowman-Brick inhibitor as well as trypsin inhibitor. So this has drastically very low levels of protease inhibitor activity in the seeds. And recently what we have done, we have taken this BBI mutant, generated transgenic mutant, and crossed it with naturally occurring KTI3 mutant, and we're able to generate a soybean line which completely lacks both trypsin and chymotrypsin inhibitor activity. So pretty much we have a line of soybean that has no protease inhibitor activity. And Lisa raised a question, okay, what is the function of this trypsin inhibitor? They are involved in pathogen protection, prevention against insects feeding. We don't know anything about how these plants will respond. Okay, we have grown these plants in the growth chamber and they are doing fine. We don't see any obvious phenotype so far. And this year, we actually planted them in the field and we should have a better idea how these plants perform. If we don't see major differences in the yield or pathogen response or insect attack, we'll be very happy, and that probably could be utilized for animal feed and as later. And time will tell us whether this is going to be very useful or not. So I told you about the major anti-nutritional factors. What you're seeing here is a 2D gel electrophoresis of soybean total proteins. And you can see there are P34, which is an allergenic protein, and you have the lectins, and also the KTI. So there are three arrows representing three different isoforms of KTI, KTI1, KTI2, and KTI3. The major the greenish yellowish spot is the KTI3. And below that, there is a circle around several protein spots, and they represent BBI. So the BBI is made up of at least 11 different genes. But transcriptional analysis of the developing soybean seed has shown out of the 11, there are only 3 specific genes which are abundantly expressed in the seed. The rest are not expressed that abundantly. So our BBI mutants have completely lacked all of this BBI activity. So with regard to the measurement of the trypsin inhibitor, traditionally, the two methods that is commonly utilized is the AOCS and ACCI method, okay? So the procedure is very simple. Basically, this is an old procedure established several years ago, and these procedures have been modified in the last 20 years because this assay, the trypsin inhibitor assay, the results of this assay is quite variable, mainly because the activity could be influenced by several factors. For example, the sequence of the reagents you add, the substrate concentration, the particle size, how long you extract the soybean meal with the buffer. So there are several variables, and subsequently, it is not an easy assay to perform. And the classical, the AOCCS method, which is called as a standard method, uses 10 ml for the assay. And it is laborious, time-consuming, and often very tedious. So later on, Lou and all modified the procedure, basically cutting down the 10 ml assay into a 5 ml assay, and also find similar results to the 10 ml assay. And again, even though the assay was cut down the volume to 5 ml, still it retains the same laborious procedure. You start with the starting material is almost 1 gram, and you extract with 50 ml, and depending on the duration, for example, the 10 ml extraction, you extract it overnight, whereas with a 5 ml, your extraction procedure varies from 1 to 3 hours. So we decided to simplify further. So we are not reinventing the wheel or anything like that. We are trying to find ways where we can simplify the procedure so we can get the assay performed rapidly and also accurately. So what we decided to do is to cut down the volume of the assays, reagents, extraction procedure, everything, so that we can do these assays very rapidly. So for example, here is an outline of the sample extraction. So I can't read anything from here, unfortunately. So the procedure is pretty simple. Basically, instead of 1 gram, we take 20 milligrams. There's a reason why we are using such a low amount of material. Because if you take 1 gram of material and extract with 50 ml, and they do it for a longer period, like overnight, for the complete extraction of the proteins, we basically speculated that if you take a small amount of volume, material, starting material, for example, 20 milligram, and use 1 ml of extraction buffer, and vortex it for a short period, like a 10 minutes of vortex, you can recover almost identical amount or same amount, equivalent amount of protein as extracting 1 gram of material with overnight extraction. And we tested this method, and it turns out that even 20 milligrams for 10 minutes extraction gives the same ratio of the protein extracted like 1 gram overnight extraction. So that way, we eliminated the overnight extraction procedure altogether, so we can actually need only about 20 milligrams of material to measure the trypsin inhibitor activity in soybean and soybean products. So I have basically provided the assay procedure. So if somebody is interested, they can actually get the details of the assay procedure on the paper I have cited at the bottom of that particular slide. So it gives you all the details regarding how the assay is performed. The assay is very simple. As I said, we did not reinvent the wheel. It's basically the old procedure, the 10 ml standard AOCS approved procedure, but we have eliminated several of the unnecessary, often redundant steps That way, what we have done is eliminated the multiple centrifugation steps, prolonged the extraction procedures, and drastically lowered the amount of reagents used. So that will will drastically reduce the cost of these assays. And also it is so easy that every lab can routinely use this. For example, the old 10 ml procedure is so time consuming, and often people used to send the samples to some different labs in order to perform this assay. Whereas with our procedure, anybody can do that in their own lab because the equipments are all you need is a centrifuge, so small benchtop centrifuges, so things will be sufficient to do this assay. Okay, again, the calculation of the trypsin inhibitor units, the way in which we calculate is also outlined. So the definition of the trypsin inhibitor unit is the amount, a drop in the absorbance of 0.1 WODI is what's our definition as one trypsin inhibitor unit at 410 nanometers. So the calculation, everything is very similar to the standard method. So there is nothing new except the volumes have been drastically reduced. The time of the duration of the extraction procedure and everything has been shortened. So now we have come up with this 1 ml assay method. So what we wanted to do is to validate our assay and compare it with the standard as well as the off-volume assay. So in order to make a meaningful comparison, we wanted to have soybean lines that shows different levels of trypsin inhibitor. As I told you, we have earlier identified trypsin inhibitor mutants, and this trypsin inhibitor mutants, KTI1, KTI3 mutants, as well as a double mutant. In addition to that, we also have generated a transgenic soybean plant that actually overexpresses trypsin inhibitor. So now we have different soybean lines showing contrasting levels of the trypsin inhibitor. And Lisa also pointed out earlier, when you knock out the trypsin inhibitor, the plants somehow compensate by overproducing the Bowman-Big protease inhibitor. As you can see in the panel C, this particular slide is a Western blot analysis using trypsin inhibitor and the Bowman-Big protease inhibitor antibodies. In the panel B, in the lane 1, is a wild-type Williams 82, where you will see the two isocymes, Krishnan KTI1 and KTI3. There are two bands in it. In the next line, which is a KTI1 mutant, the top band is missing. The KTI3 is present. On the next lane, you will see the KTI1 is present, but KTI3 is present. And in the last lane, which is a double mutant, there is no KTI protein is present. So it's completely gone. But when you look at the Bowman-Big accumulation, you see a drastic increase in the accumulation of the Bowman-Big protease inhibitor because the plant is trying to compensate for the absence of trypsin inhibitor. So that tells me something, that these proteins might have some physiological function. So it may not be that desirable to get rid of all these proteins. But as I said, the time will tell us because we are growing these plants in the field this year. So now we have soybean lines with different contrasting levels of trypsin inhibitor. So what we have done is measure the trypsin inhibitor activity using 3 different methods, namely the 10 ml method, the 5 ml method, and the 1 ml method. As you can see, the values are very, very similar. The trend is identical to each other. So basically, whether you're using the 10 ml volume or the 5 ml volume or the simplified 1 ml volume, the result is going to be pretty much consistently very similar across both. So we were happy with this procedure, and we decided to look at the trypsin inhibitor levels. No, we also tried to see whether we can even further reduce the assay volume to 200 microliters. So we developed a microtiter assay. So here, what the volume is, total volume is about only 200 microliters. And we get the similar trends, but the number, if you compare to the 1 ml and 10 ml, is slightly lower. That basically needs to be a little bit playing around the conditions to get back to the same levels as the 10 ml and 5 ml. But what this slide indicates to us, that it should be possible to adopt our technique to the microtractor plate, so the plant breeders, where they will need to screen hundreds and hundreds of samples, they can get a rapid method to detect for example, if they are looking for a mutant, they should be able to easily screen for them. So the next step is to look at some of the soybean meal, the commercial soybean meal. And here I have listed about 10 or 12 different soybean meal samples, and we compared the activity, trypsin inhibitor activity performed by the 10 ml standard MUST procedure with our 1 ml simplified procedure. As you can see, the values are very similar. They may not be exactly identical, but the trend is very, very similar. So we are very confident our 1 ml simplified assay procedure is quite useful, and it can be extensively, widely applicable for measuring the trypsin inhibitor in soybean products. So the question is, if you look at the soybean meal, the commercial soybean meal, the trypsin inhibitor activity is drastically reduced. So the question is, what is happening to this trypsin inhibitor? Is the protein still present, or is there anything happening to the protein? So in order to demonstrate what's happening to the KTI protein, here what we have taken is the soybean proteins from a couple of commercial solvent extracted denatured soybean meal and a couple of extruded soybean samples. And the first lane is the William 82, which is a wild soybean cultivar. And again, we are doing a Western blot with KTI antibody. As you can clearly see, KTI is present in all the samples. What this suggests is that even though the enzyme activity is drastically reduced, the protein is still present. So the heat denaturation basically inactivates the protein, but it still retains the protein by intact. And then we obtained numerous samples from industry partners. And at this point, I should thank Philip Lobo for his effort to get these samples from several industry partners. And we have more than thousand soybean meal samples from all these industry partners. And we actually have looked at most of these samples. We still have some samples to be analyzed. And I'll present you the results from the analysis of these samples. I would say out of 500, this particular slide shows you the trypsin inhibitor activity measurement of 531 samples. And these are all commercial soybean samples obtained from industry partners. And you can see the trypsin inhibitor about, I can't exactly read it, it's about 200 and whatever, the first green bar has less than 4 trypsin inhibitor units from 0 to 4. And the next one is anywhere from 4 to 8 total. And you have a few samples which is above 8 units. And the ones with values above 8 are mostly either extruded or expelled soybean or organic soybeans. And so the processing industry has done an excellent job where they have regulated the trypsin inhibitor content and at acceptable levels, in my opinion. Okay. So, as I told you, you can, out of the 531 samples we looked at, they can fall in 3 different groups. One between 0 to 4 trypsin inhibitor units, the other one 4 to 8 units, and the ones above it is above 8 units. As I said earlier, the processes have handled this very well. Most of the trypsin inhibitor units are significantly lower. And also, we also tried to see whether there is any correlation between the protein concentration and the trypsin inhibitor units. And here we have not taken all the 531, the results of the 531 samples. This one is probably only about 80 different samples. And when you plot it against the protein concentration and TIU, there's a linear correlation, but this correlation does not apply if you include the extruded and samples, because the extruded samples are way off with this. Yeah. So this slide basically summarizes what I have been saying, the advantages of using the simplified method. Basically, this method is easy, fast, and also reliable and reproducible. And you can drastically reduce the cost of the assay, and every lab can actually do it in their own lab. So with that, I would like to acknowledge some of the people who are involved in this work, my lab members, as well as funding from United Soybean Board and USDA-ARS. Thank you very much.